Hi, good evening, and welcome to the ZP Developer Zone. So we like to do this Developer Zone once a week, and I think you'll probably see on my slides shortly that I've got the date down for the 14th of July, but admittedly I'm recording this on the 13th of July. I'm just starting to find it's quite convenient to be able to come on and do this earlier, just because it's Saturday evening and there's no emails, there's no work, so there's a fun way of spending Saturday evening is doing this quick video. Things I'm going to talk about this evening is lactate sensors. So, we do get a lot of inquiries around lactate sensing. And in fact, I just want to change cameras for a second just to kind of say that it's not all going to be talk. I do have some hardware in front of me. So, in fact, I might even run some of this hardware this evening as well. But it's not all about talk. I'll do a quick show and tell, but it may not be as much show and tell as I would normally do. The reason being is we've actually done something like, I don't know, 4 webinars this week, and we've actually have demonstrated the technology quite a bit in there. But I do have some wearables in front of me, and I also do have some what we call IVD, in vitro diagnostics. So I will be doing some of that shortly, and so let me just jump back to the main, let's say, theme now. So what I want to talk about is lactate questions. People are very interested in wearable lactate sensors, and I'm quite happy to talk about that. And we have put lactate sensors onto both what we call 2D and 3D structures. So we do have 50 patents over a wearable watch format using microneedles, and that's something I can touch on. Somebody's asked us about hollow needles within our patent portfolio. We do actually have a lot of IP. Oh, sorry, we do have IP around hollow microneedles. Just be careful about hollow microneedles. I think something that people don't really think about a little bit is, do they have a freedom to operate? So obviously, if you're going to do something, especially in the kind of medical space, it can be a very litigious space. And so a freedom to operate is quite important. Do you actually have IP to defend what you're about to do? Because if you're going to do something, let's talk about CGM, continuous glucose monitoring. This I'm holding up to the camera at the moment is it's a technology from Abbott. It's not Zimmer and Peacock, so let's be very clear about that. But behind this, there's a lot of IP, and obviously, Abbott will have invested a lot of a lot of money in this IP, and so they're rightly going to defend it. So you've got to be very careful about what you do in the medical diagnostic space. So it just came to me just now as the term hollow microneedles came up that actually there is a lot of IP out there. We've got a little bit of IP around hollow microneedles, but be careful about actually what you're doing, because if you haven't even considered the term freedom to operate, definitely consider it. Number 3 is about sweat analysis. At ZP, most of our experience in sweat analysis is around lactate and potassium. And also the next one here about immunosensors. So somebody is working on a immunosensor type product, or at least project, for something that we call geosmin. So I will be coming back to this technology in a bit. This is also very relevant to question number 5 as well. So this is not a nice thing, but this is something that's happening, that people are having their drinks spiked, maybe in a social situation like a bar, and people are kind of, things are happening to them when they're unfortunately under the influence of these drugs that have been spiked into their drink. So I do want to discuss that, and can we actually make a sensor for that kind of technology? I think the quick answer is, let me just change cameras for a second. At ZP, we do have that kind of technology. We have apps that make Bluetooth connection with meters, and in fact, this is available for sale on our web store, this platform that is, and you can basically, let's say somebody had a spiked drink, you could just take, 50 microliters, put it on there, and we could run a quick assay on that and try and discover what actually is in that, or any particular compounds, or specific compounds rather, are in that sample. Now, the ZP platform means that you can buy the hardware, you can buy the screen printed electrodes, but you then have to develop the specific, how do I put it, the specific test itself. So there's still, some science to be done. So we do have a platform for people who want to create new applications and want to be able to do proof of principles, etc. So I will come to that as question number 5. The reason I'm not going to do too many demonstrations this evening is because actually we do a lot of online webinars. I'm just putting a password into something. We do a lot of online webinars, and I don't think I need to be repeating them this evening in terms of demonstrations, but on the 7th of August we do have a wearables webinar. I appreciate some of the questions that are in this evening. You've already attended our wearables webinar, so I wouldn't certainly ask you to do that again, but if you haven't attended one, please attend one. We delve a lot deeper into wearables in that. I'm wearing a wearable microneedle watch here. Oh, sorry, I should say as well, if you see a link pop up, those links will be put underneath the video as well, so that will help you navigate around a little bit. On the 31st of July, in fact, well, let me have a look here. So on the 7th of August, we're doing an IVD workshop, an in vitro diagnostics workshop. So if your kind of interest is IVD and point of need testing, I sometimes call this spot testing or discrete testing. And it's literally that situation where you have a sample, you put the sample on a sensor, you get a result, and the sensor's done with. That I would call a discrete or a spot test. So that's what we'll be doing on the 7th of August. On the 31st of July, so not that far away, if tonight's the 13th of July, just a couple of weeks, we'll be talking more in depth about wearable sensors there. We always do 2 webinars, one in the morning, one in the afternoon. We're pretty inclusive. We understand that, you know, some people are interested in these topics and they're in Asia, and other people are interested in these topics and they'll be in the Americas. So, you know, we do a couple of webinars a day to make sure that everyone in different time zones is able to attend. If you're in academia, we do a webinar particularly for academics or people who are interested in translating, and that'll be on the 18th of July, so that's just 5 days out from now. Again, 2, 1 in the morning, 1 in the afternoon. If you see a link pop up, I'll put the link underneath the video, and that should help you navigate. So question number 1 is about lactate. The thing about making wearable biosensors that are going to be used on human beings, I think you have to. You're either going to do it or you're not going to do it. You have to have the funding to be able to really do this. I sort of have a slight feeling that when you don't have the funding, you tend to cut corners, and you can't really cut corners when you're doing medical diagnostic development and it's intended to be used on humans. You could cause harm to humans. You could cause infections in people. You know, you've got to be really cautious. So it does concern me a little bit if I feel like people are not really properly funded to be doing what their ambition is. I did see something on LinkedIn the other day that says, you know, well, congratulations, you've raised $25,000. That should be being now used to raise the next $250,000. Congratulations, you just raised $250,000. That should be used to raise the next $2.5 million. So it's really like, you know, when people have small sums of money, they think, oh, therefore I need to develop a bit of technology. No, you need to be actually really figuring out how to really raise the money. I mean, if we're talking about the cost, so for example, this watch has had something like 20 million euros invested into it. It's got something like 50 patents behind it. That's, you know, it's not low cost to do this kind of work. I'm thinking about BioLink down in San Diego. They've raised in the last 4 years to do a similar technology where they have microneedles. Their microneedles are worn here. They've raised 158 million US dollars in the last 4 years. So be really careful. I mean, it's a bit concerning when people are going into programs where they're not really properly funded, because I think it can lead to corner cutting, let's say, and that can be dangerous, especially if you're dealing with putting sensors on people. So somebody's trying to develop something, and so I suppose that's it then. They're trying to develop something for monitoring interstitial fluid. It's really come to the wearables webinar, but there's there's lots of ways of doing interstitial fluid. When you look at a product like this from Abbott, and this is not Zimmer Peacock's technology, but it is a nice illustration actually, and I wasn't intending to show that for this case, but that's a wire sensor, you know, and so Abbott are sampling the glucose in the interstitial fluid using that kind of wire. We're similarly got that kind of technology, but we're also able to sample the interstitial fluid using something like a microneedle. So this is a patch that we have that kind of gets worn on the arm. The watch strap helps to pull it through into my interstitial fluid. So that's what they want to do. They want to measure lactate and interstitial fluid. You can build prototypes with products that Zimmer and Peacock has on its web store, but I do not want people to be using these products on human beings or on animals. If we put stuff on the website under our terms and conditions, you'll see that it says very clearly sales terms and conditions. Don't ask me for our sales terms and conditions. Just Google Zimmer and Peacock sales terms and conditions, and you will find them. That this is for research purposes only. Zimmer and Peacock, we're ISO 13485. So if we're going to be getting involved in products that are actually going to end up on human beings, then that's an ISO 13485, and you have to be able to afford to be onboarded by us and go through the proper, let's say, procedures, and also paperwork as well. I mean, once you start designing for human beings, it's super. When I say designing for human beings, when you're trying to make something that's wearable for people, you've got to really do it properly, and all the documentation's got to be in place, and it doesn't come cheap to do that kind of work. So you can buy materials off us to make prototypes, but our sales terms and conditions will say for research only, and that doesn't mean clinical research. I mean, that clinical research, animals or human beings, certainly not. You can build a proof of concept feasibility prototype. Just means, for me, could mean TRL3, something that you technology readiness levels at number 3, something that you could do in a laboratory in vitro, not in vivo. Our lactate sensors, whether it's the 2D lactate sensors that we have, which you could describe on a screen printed format, or we got 3D formats. We got lactate sensors on wires, for example. This is not our product, but I do realize we do have something on the website where we have lactate sensors much in this kind of wire format. Either way, they function, they work. Yes, certainly they do. So theoretically, you can produce a prototype, but we're not putting those things on the website are just for really doing proof of principle, and not for putting onto people or into people. That would require a very different kind of engagement with Zimmer and Peacock. But if you need to do a benchtop test, then they would be fine. But to really collaborate with Zimmer and Peacock, we won't support we'll sell materials to people on the website, and if they're skilled enough or have the right kind of background, then they can use them. But if they require deep support from Zimmer and Peacock, then we would rather people didn't buy those products, because in order to have a proper engagement with ZP, with us answering questions and troubleshooting for you, we need a proper budget in place that we can actually become involved. So we rather people didn't, unless you're really funded to do wearables, my advice would always be don't do it. Do something like in vitro diagnostics, but don't try and do wearables on a low budget. We do have a lot of lactate technologies. Just recently we put some better pages up about this, so we do have some URLs here. If you see the URL in this presentation, and I'll also put the URL underneath the video as well, and that may help you navigate. So you can put together, hopefully using some of those URLs, prototypes, but those prototypes should only be really bench testing. And if you need Q&A with Zimmer and Peacock, or you need Q&A or support, or you have questions, then you need a proper budget in order to actually, so that we can actually put some attention on it. If you're a strong academic and you understand the technology quite well and you can put it together, fine, go ahead, but it's only for bench testing, let's say. So you can buy products off our website. The couple of links earlier on will help. But we won't be bringing in our expertise. And our expertise, I realize, that we are, for example, working on embedded sensors, and these are going into animal studies at the moment. It looks like a joke, but both pig and fish are things that we're actually working on at the moment. And we have done lactate sensors on people. This particular image is of video, but it's not something that we won't or can't collaborate in some deep, meaningful way, unless there properly is a budget actually in place for ZP. The engineers, we've got really strong engineers, but and really good facilities. And actually, we've got a patent family of like 50 patents and etc. So there's a real cost, to ZP, and that's why we sell things on the website. You're welcome to buy them. If you're able to use them, that's great. But if you do need our deeper involvement, then we would require a budget for that. Question number 2 is kind of related a little bit to question number 1. Another way of getting interstitial fluid out of the body. In our particular technology, most of our experience is actually functionalizing either microneedles or functionalizing these kind of filaments in order to actually essentially put the enzyme in place and measure things like lactates, glucose, ketone, essentially in vivo. But there is some of our IP also does cover hollow microneedles as well. But we don't have any hollow microneedles available on the web store. And if people do want hollow microneedles from ZP, then it's going to be a considerable program with us. Because you're going to need those hollow microneedles to be put onto human skin and basically bring the interstitial fluid out of the body. And think that as soon as you get into the world of breaking the skin, at a minimum you could cause somebody an infection. So these things have to be done very, professionally, and you have to have the right ethical approval, etc. So we don't want to become involved unless there's a proper budget in place and it looks like you can act in a kind of ethical, responsible manner. But we do have a lot of IP around the use of microneedles at ZP. Now, something else that we've been involved in quite a bit is actually, we have been involved in is sweat analysis. I think most of our experience is actually in potassium and in lactate. So people want to take our biosensors. We do have a lot of biosensors. It's always important. It's always interesting to kind of know what people do want to monitor in sweat. But we do have technology for doing sweat analysis. I would suggest that the ethical approval for sweat analysis is a bit easier than transdermal filaments, or easier than microneedles because you're not actually breaking the skin. But even if you do phases of this, there needs to be the right kind of funding in place in order to do this. Now with sweat, we do have some data out there that we have done lactate monitoring, measuring people for their state of exertion. Here we had a sort of sweat patch on somebody, and we were able to monitor the lactate when they started to strain, and we were able to compare ourselves against academic colleagues, and we got really good sort of correlation. And you can buy our technology. You can essentially play with it, but this will really bring you to sort of a proof of principle in the lab. Putting this stuff onto people and actually, getting meaningful data out of people is much tougher. And ZP won't just support, I bought a lactate patch off you and I've put it on somebody and I want you to help me understand and calibrate the data. No, there we will actually require a proper program with people. But we do have parts, or let's say a toolkit. We've got lactate sensors, we've got sweat patches, we've got electronics. So if people feel like they can read what's online, they can follow what's being said there, and they're competent, then they can have a go at putting together proof of principles for doing lab experiments. But again, just as an FYI, if there's a few companies out there that sort of sell into the R&D market, but when you really read their terms and conditions, you'll often find that they say for research purposes only, and actually they're not therefore intending you to make a pharmaceutical and give it to people, for example. Well, with us, we might have pieces on the website for research purposes only, but we have to be very careful that it's for research purposes only, because you don't really know what people are going to necessarily do with stuff that you sell. So our terms and conditions, don't ask me for them, as I say, just Google Zimmer and Peacock terms and conditions or sales terms and conditions. You will find that what we're selling is for research purposes only. We are definitely doing technology that actually is going on to people, going into animals, etc. But those are proper programs. Those are not just web store purchases, for example. But we put stuff on the web store because then people can put together a prototype. They can show it to an investor. The investor says, okay, yeah, you guys look like you're competent, and then they can raise more money. So we will put materials on the website for setting up, as I say, a proof of principle. I think generally, we're imagining that somebody needs to demonstrate something to an investor, but you can only really show a proof of principle. You can't start putting sensors through the skin as a proof of principle. You have to be really kind of funded to, you have to be able to answer questions about sterility, etc. And you've got to be well funded to be in that kind of domain. But if you want to do something in sweat, then we do have sweat technologies on the website, and you can put together the system, but it's really only for doing sort of lab testing. Just out of interest, we did answer a question a few weeks ago now. I'm just going to question number 4, which is about geosmin. Geosmin is a sort of biomarker for bacteria. It can lead to problems in things like salmon farming. And it's quite nice because we were talking about, you have to make an immuno-type sensor for that. And there was a question that kind of came up, which was, how many molecules per centimeter squared do I need? So if you're trying to make an immunosensor, you generally have some sort of antibody. These days, people are starting to use oligomers, azos, and you need a certain density of molecules for this to kind of work correctly. So a bit of a starting point, if somebody's not sure, is you'll probably need about, let's talk about oligomers. You'll need about 1.5 10 to the 12 molecules per centimeter squared. Our electrodes are approximately a third of that, so you'll need a third of those molecules. But the density stays the same, rather. That the number is 1.5 times 10 to the 12 molecules per centimeter squared. This is in, if you're trying to make an immunosensor, for example. I do have a reference here. I'll put it underneath here, and we have talked through quite a lot about actually how to build an immunosensor or an immunoassay. But the one question that's probably not answered a lot in those webinars is how many actual oligomers that you may need, and so that's why I wanted to put that 1.5 times 10 to the 12 per centimeter squared out there. But these slides that I'm going through quite quickly, we definitely do have quite a lot of material out there, and I've been through these slides quite a few times, so I don't want to spend the time there. Maybe the reference that I will link underneath the video will be helpful, and then if people get stuck again, then maybe I will return to it. This is something that this is happening. I mean, I don't want. Basically, what's happening is people may be in a bar situation or some sort of situation that's social, and they may have a drink, and the drink might get spiked, and the person is kind of then abused underneath the influence of that spiked drink. And so they want to sort of come up with a way of actually detecting molecules in spiked drinks. Now, basically, I mean, this is a lot of what we're very interested in at Zimmer & Peacock. So I'm just going to give you, we do have a platform that's called Sense It All. It's an IVD platform. In fact, I've got the platform in front of me at the moment. I've also got the app here. You'll see me use this app in a little bit. At the moment, the app's looking for what's called a QR code, and that QR code will become significant sort of nowish. We have an online database called Julie. So this is important because if you're trying to do the development of, for example, spotting opiates that might be spiked into somebody's drink, we have the ability to be able to process that data and turn electrochemical signals into concentrations, and we do it in online in a database called Julie. But Julie works with the app, so I will show you this app very shortly, and that app actually controls the hardware called Sense It All. So Sense It All you can get, so first of all you'll probably need to register on Julie, open up an account there. When you've registered on Julie, you can actually then install an app on your phone. If you've got an Android, use this QR code. If you've got an iPhone, use this QR code. You'll find that both those apps are on those various stores. And when that's installed, you can actually find, I'll just do this real quick actually. At this moment, I've just scanned that QR code, and now my phone is ready to do an amperometric experiment. And actually I can connect my phone to the device that I've got here. When I say the device that's got here, I'll just put my hand on it real quick. So this is the Sense It All. I can Bluetooth connect with my phone, suddenly, by the way, this technology is also applicable to our wearables as well, but I'm talking specifically about IVDs at the moment. Makes Bluetooth connection with my phone, and I can start driving it. I'll just go back to my screen for a second. So we've got amperometry, we've got open circuit potentiometry, we've got electrochemical impedance spectroscopy, we've got linear sweep voltammetry, we've got cyclic voltammetry, and we've got square wave voltammetry. So if you're interested in looking at drugs of abuse in or used for spiking drinks, then I suspect it'll be linear sweep voltammetry, cyclic voltammetry, and square wave voltammetry will probably be what you'll end up using. I suspect most likely square wave voltammetry. These QR codes, these QR codes on this app, we've got the ability to read new QR codes. So I'm just going to scan that for a second. So I've just scanned that QR code. So this app is the capability to be a genuine potentiostat, doing amperometry, open circuit potentiometry, linear sweep voltammetry, impedance spectroscopy, square wave voltammetry, linear sweep voltammetry? So you can do genuine electrochemical methods, but very quickly you can also do targeted analytes. And so these different QR codes, if you open that Julia account that I recommended and install that app, you're going to start understanding that actually we can very quickly go from electrochemical methods to final analytes, because actually we have a very capable cloud system that can crunch the data and turn that data into meaningful concentrations. So these are, for example, QR codes that people can play with. So we can go from these kind of methods of amperometry, open circuit potentiometry, EIS, linear sweep voltammetry, cyclic voltammetry, and square wave voltammetry, actually go from basic science to something that you can actually, test and analyze. So, this could be we could have a QR code for an opiate or a tranquilizer or something along those lines. So I'm going to put some links to the starting system here, so you can get the hardware and you can also get the screen printed electrodes. So we suspect that it's the development of the assay which is what you're left to do. You do have to develop the assay, and that is what's going to take your skill. But otherwise, the hardware does exist for this kind of thing. So lactate question. We have to be really careful about getting pulled into programs on wearable sensors, especially when the word interstitial fluid is there, because you can cause harm to people just inadvertently, by giving them an infection, etc. So we do we definitely will not be casually drawn into a wearable biosensors unless somebody's really well funded, because otherwise you find that people are actually cutting corners and it can get a little dangerous, let's say. Hollow microneedles, we do have some IP around hollow microneedles. We have nothing available on the website as such. And again, I think hollow microneedles kind of, be careful, because you've got to have a freedom to operate in this kind of space, which means you probably need patents. So you need to have checked out what the patent landscape actually is. Sweat analysis, we do have kits on our website for sweat analysis, but really we're not intending you to put those on the people and off you go. What we put on the web store is just to get you going, to do a proof of principle, something to impress with a lab experiment, impress a investor or an investor that you sort of know what you're doing. But the equipment on the website is not then intended to be kind of your product and you can just go to market with it. We are ISO 13485. On our ISO 13485 certification, it says contract developers and contract manufacturers of biosensors. So that means something. That means that we'll put technology on the website, it shows capability. It also means that our academic colleagues can do some sort of lab experiments. But if you're really commercially orientated and you want to go to market, then we do need you to engage us on properly funded programs. With the geosmin, this is easy. It becomes a little bit easier once people are coming away from wearables. Wearables, you're making contact with the skin or you're breaking through the skin. That's a different risk level. When you're starting to do things like in vitro diagnostics or point of need testing, that's a lot easier in my opinion, from a regulatory perspective and also a risk perspective as well. So if you're trying to make immunosensors, and they're IVD immunosensors, and a lot of people are starting to use aptamers. That was the term I was trying to look for earlier on. It wasn't oligomers as such, it was aptamers. Then you need 1.5 times 10 to the 12 molecules per centimeter squared is a good starting point. That has to be optimized, but at least you know where you have a starting point. And I think this illicit drug spiking is something that we've actually, been really interested in ourselves. And I genuinely think we actually have the right kit to do this. So what I mean by that is, so we have sensors. Let's say we can take a sensor. I'll just click that in there. There's a lot of this is, by the way, is not powered by a PC or anything like that. It's low power. In fact, yeah. Then we'll put a sample on here, which I'll do now. Thinking and doing at the same time. Put a sample onto that, and then what I might do actually is just start that off. In fact, I'll just make a Bluetooth connection with it. Yep, make a Bluetooth connection. Done that. I'll just put some names in here. So I might even put Spike here. Spike. And off it basically goes. And then it will just run. So I have really no problems with the idea that you can make that kind of technology. And actually I think if ZP was doing this, we would do it quite well. Now I appreciate that this might be more of a kind of college level program, and that makes it harder because you're basically learning at the same time. But with a team like ZP, we would have done that quite quickly. But probably my biggest, let's say, I won't say problem as such or concern would be the business model around this. Just because you're capable of doing it doesn't mean that even if we did do it, it would actually turn into a business as such. But if people are interested in doing this and doing it in a commercial sense and they feel like they're funded well enough, then I think it would be something that we would be interested in discussing. So I want to say thank you very much. Don't mean to be down on people trying to do wearables, but wearables is pretty tough. I mean, I think I did hear the other day that Dexcom, that makes one of the CGM bigger players, they may have spent $500 million doing their continuous glucose monitor. BioLink, I've got more up-to-date data on them because it's in the public domain. They've raised about 158 million over the last 4 years. I think even prior to that, they've been around for 7 years doing things like SBIR grants. So it's a worry when people come and they're trying to do it on a low budget. You can buy technologies of Zimmerpeak in our website, but our terms and conditions say they're only for research purposes only so you could do a proof of principle, and that proof of principle really means lab stuff. And if it's not clear enough on the website that you can do that, so you can understand the technology, you can just get on with it, then you probably need us. But that is a program, and we would look for people with the correct level of funding in order to do those programs. I think I'll finish it with that. But if you've got any questions of ZP, don't hesitate to reach out to us. Also, do know that we do have wearable webinars, point of care webinars, and also webinars specific to academics. If you've got any questions, don't hesitate to reach out to us. Okay, thanks very much.